Okay. Okay. So the jumping genes, it's called the transposones, right? Uh, Means it can jump to the any location of the DNA, but it is restricted to the one cell only, right? Within the cell, the DNA can be jumped. That those DNAs are called as a transposon, or those DNA or the genetic, the part of the genetic material, it's called the transposons, right? So what here you can see, uh, the red part can be jumped to somewhere else, right? The green part can be jumped to somewhere else, right? So transposons are sequence of DNA that can move around different positions within the genome of the single cell. The donor plasmid containing the transposons co-integrate with accepted plasmids. They can replicate during co-integration. So if you have the two plasmids, right, you must be knowing that there are several copies of the plasmid in one single bacterium. That is not only the one copy of the plasmid, and it varies microbes to microbes, right? It varies organism to organism, and it varies strain to strain, right? If E. coli is the different strain, so we can say the different capacity of the plasmid is there. And if we talk about the staphylococcus, if we talk about the some different strains, the plasmid copy number varies from strain to microbes, right? So the transposon are the elements that can be jumped from one plasmid to another plasmid, right? Okay, the link will be closed. Let me remind you all that at 12:30 I will close the link, right? So you have to come before 12:30 and you have to fill the form. Otherwise, it will be marked as absent, because see, in between I cannot take your attendance, right? I have to complete the syllabus as well. Okay. So both plasmids then separate and each contains the resistant gene carrying the transposon. So whatever the transposon we can or they can transfer, they have the R gene. It's a resistance gene, right? So now both the plasmid, if one transposon jumped to the another plasmid and they are having the R gene, so now both plasmid can have the R gene capacity. Okay, so now the here you can see the transposon. It shows in the red color, right? Okay, in the next slide, you can see the plasmid come inside the cell, and it has, now you can see, integrated into the chromosomal DNA, right? Into the cell genome. Okay, and now you can see the green part, that is, it can be detached, right? Okay, so now you can see the transposon red is attached, and that green part from here it moved to here. Okay, now you can see the sum of the red part, it is again separated. Okay, now the red part, again you can see it is attached to somewhere else, right? So this is the transposon element. First it binds, it integrate, then it attach. This green part, it directly jump to the another location. Okay, now you can see the two different plasmids are carrying the one red transposon and one green transposon, right, which have the R genes, right? So here you can see some of the part that is, we can say, red and green. So let me go again so you can see the exact procedure. See now, the transposon is totally two different color of transposon. Here you can see the red part and the green part. Now in the next slide, you can see the plasmid come inside in the cell, and here you can see the red part. It is going to integrate, right, in the cell genome. Now the next step is the green transposon is being jumped to somewhere else. Now you can see it will integrate at at another site. Now the red part is, now it will detach and it will integrate to some another site. You can see over here, right? Now both the plasmids, they are having the different transposons, red color and green color. They are separated and they are carrying the R genes, right? Okay. So now here you can see A, B, C, and D plasmid, right? So B is the donor, right? Here you can see transposon donor. So here you can see the A part and this is the B part. Now B, it will give to the A, right? Because B is the donor, A is the acceptor, right? So B will give some of the part to A plasmid. Now you can see the exchange of the genetic material, and now A is having the some transposon elements. Okay. So what is the mechanism of resistance gene transfer integrons? So uh, if we talk about the mechanism, now here you can see the integron is the part, right? It's the mobile DNA and it has the multi-drug resistance genes, right? And it cannot transfer by its own, right? They cannot get self transfers. They need the help of the plasmids, right? So each integron, is packed with multiple gene cassettes, each consisting of a resistant gene attached to a small recognition site. Here you can see the integration has the cassettes. Cassettes means the segment of small genes, right? It's called the cassette. Say, for example, you have introns, exons, exons, just like that different package of the genes, it's called the cassettes, right? And this integron, it is made up of different cassettes and it can integrate into the specific plasmid, right? So these genes are encode several bacterial functions. Now what is the function of this integron? So the genes present in that integron as a cassettes, right, it has a different functions. What are that functions? It will give the virulency, right? So it will give the virulency, means it may be pathogenic, right? Or it may give the resistance to the antibiotics, right? There are two different types of functions, but yes, once it get integrate, then only it gives this type of functions. This portion is called the integron, right? Okay. Biochemical mechanism of antibiotic resistance. What is the biochemical mechanism? Why antibiotic resistance is there? What happened actually at biochemical level? So prevention of drug accumulation in the bacterium. So uh, actually drug can't penetrate into the bacterium, right? It prevent drug. So what prevent? There are cytoplasmic membranes, there are cell walls, right? So something is there that can prevent the drug to penetrate into the bacterium, first step. Then modification, protection of the target site. If it is the 30S ribosomal site is the binding site for the drug, 30S can be modified. What is this happening? I don't understand today. So it can bind the 30S or 50S site, right? So definitely it can't get bind exit, it can't get the proper site to for the binding, right? Use of alternative pathways for metabolomic or growth requirements. What is the use of the alternate metabolic pathway? See, in our body there are different types of metabolomic pathway. What are those pathway? If we say TCA cycle, glycolysis, scrap cycle, and certain amino acid biosynthesis, nucleotide biosynthesis, or so many pathways are there, right? Gluconeogenesis or some. So they all are the metabolomic pathways, right? So what happened? Uh, they generally in the normal bacteria, they have the different metabolomic pattern. I'm talking about our metabolomic pattern. So if we talk about the bacteria, the metabolomic pathway what they follows routinely, in the case of drug resistance or antibiotic resistance, they follow different metabolomic pathway. So the same pathway, same channel will not be followed for the growth requirement. So your drug will not be working over there, right? So again, it leads to the genomics by producing an enzyme that inactivates antibiotic. Yes. So bacteria are producing two type of metabolites. We can say primary metabolites and secondary metabolites. So primary metabolites, say for example, is a glucose, right? In the lag phase and at the initial log phase, bacteria will produce the glucose, but that glucose can be used as a source of their growth, right? So it is the primary metabolites. Always primary metabolites can be utilized by the bacterium. And the secondary metabolites always it will be produced at the stationary phase, and definitely that secondary metabolites can't be useful for the bacterium itself, right? Say for antibiotics, say for enzyme, it will be useful for another thing. And due to the secondary metabolites, the pH will be changed and everything will be changed, right? So bacteria produce certain enzymes that activates the antibiotic. So already whatever the antibiotic you are applying, but bacteria can deactivate that antibiotic. Now quorum sensing. Quorum sensing means quorum sensing is the group of bacteria, they are talking each other, they will cross talk each other. It's called the quorum sensing. Say for example, if you want to make a biofilm, right? So biofilm, that is the film derived by the bacteria in one layer. So bacteria will produce certain molecules. Always bacteria will talk, right? Obviously, you must be thinking, what's this? Bacteria can talk. Yes, bacteria can cross talk. They can produce certain molecules, certain inducers. It's called the autoinducers, right? And they can convey certain messages to another bacteria, right? And it's this talk is called the quorum sensing. Means bacteria can sense the things, right? So this quorum sensing is actually, it will affect the antibiotic resistance. So what happened actually during the quorum sensing? Let me teach you in the next slide. Okay. Okay. Antibiotics normally enter bacterial cells via porine channels in the cell wall. Cell wall has the porine channels, right? Different porine proteins are there in the cell walls. You just go for the gram negative cell wall structure and gram positive cell wall structure. You will get the porine channels. So antibiotics enter through the porine channels. But if porine channels is blocked somewhere, right, or it doesn't allow antibiotic to come enter into the organism, so it won't, right? So permeability will be increased during the resistance. So that is the thing, that antibiotic can't enter into the cell wall. Now porine loss, totally porine channel is lost. New porine channels in the bacterium cell wall do not allow antibiotics to enter the cell wall, right? Now here you can see, here bacteria can easily enter, but these are the new porine channels. So bacteria it can't get allowed to enter in the new porine channels. The structure is totally different. Efflux pumps. See, bacteria having the different transporter system. We can say ABC transporters, right? ATP binding cassettes. It's called the ABC transporters. Here you can see the ATP binding cassettes, means ABC transporters, means whenever the anything want to enter into the bacterial cell, or from the cell, if anything, any material, any molecule, if they want to go outside of the cell, they need the help of the ATP, right? Without ATP, they can't enter, they can't get out, right? So these are the ATP binding cassettes. With that cassettes, it will bind and it will go inside and it will come outside, right? So it's called the ABC transporter system in the cytoplasmic membrane. So cytoplasmic membrane have certain proteins. That protein will help antibiotic to come inside and to go outside. But this ABC transporter they are not working anymore, and they will not help antibiotic to come inside. So efflux pumps will not be working. So this is the major mechanism for resistance, especially in the tetracycline, right? Whenever you will use the tetracycline, the ABC transporter system will be lost, right? So cytoplasmic membrane transport proteins will not help antibiotic to come inside, and especially in gram-negative bacteria, inhibit the plasmid mediated synthesis of porine channels, right? It will inhibit in gram-negative bacteria, porine channels will not be synthesis, right? Which obstruct the influx of the hydrophilic penicillin, especially ampicillin. And in case of penicillin and ampicillin, porine channels is the responsible because synthesis will be inhibited. Now, when antibiotic target site will be modified structurally, so antibiotic can't able to bind, right? Here you can see the target site of the antibiotic, right? This is the antibiotic and this is the target site. Easily it can get bind on this, right? But if you see over here, the modified target site, you can see the square. Now antibiotic can't bind. So if it can't bind, it can't penetrate into it and it can't get inside to the cells, right? Interior of the organism. So here you can see the antibiotic target site may be modified, right? On the bacterial cell surface, on the cell wall, on the 30S, on the 50S, whatever the binding site is, irrespective of the binding site. Okay. So modification and protection of the target site, resistant resulting from altered target site. So what are the target sites? If we talk about the target site, so first that is the cell wall, right? You know the cell wall, there is attachment site or target site. Then ribosomal point mutation, we have in the bacteria 50S and 30S. Generally, the antibiotic will bind to 30S. If the mutation is there in the 30S, the sequence will be changed. So the sequence can't get recognized to that specific antibiotic, right? So point mutation is there, so antibiotic can't able to bind it, right? So which are this antibiotic that can not able to bind at the 30S or 50S? So it's tetracycline, macrolide, and clindamycin. Then DNA gyrase. DNA gyrase, that is the enzyme, will be helpful during the replication. So it will unwind the DNA. It will create the DNA fork, right? Now DNA gyrase is already altered. So your antibiotic will not bind to DNA gyrase, right? So what happened? Your replication will be carry on. Your bacteria will be live longer, right? No antibiotic is there. It will not work, right? So fluoroquinolones, furo, fluoroquinolones, right? Modified penicillin binding proteins, streptopneumonia. Especially penicillin binding proteins are there, right? So it can uh, uh, it can alter the binding of the penicillin. So especially for the penicillin, for the rifampicin, mutation in DNA dependent RNA poly. Now RNA polymerase, it is responsible for the transcription and all this thing, but RNA polymerase is mutated, so no more rifampicin can bind over it. So transcription is there, translation is there, means your bacteria can get doubled in the proper timings, right? So here rifampicin will not work. Antibiotic inactivation. Now, how antibiotic will be inactivated using the enzymes? Now, as I said, bacteria is producing the secondary metabolites and it can produce enzyme as a secondary metabolite, right? So what happened? Here you can see the antibiotic is there, right? And your enzymes are there, right? So now your enzymes can inactivate the antibody and it can't get bind. Here you can see antibiotic. This enzyme will attach to the antibiotic. So antibiotic will not able to bind, right? Now it's not able to bind to the target site. See, and sometimes what happened, certain different types of enzymes can kill them antibiotic, right? Or totally it is modified the structure. See, here you can see the structure has been modified. Here you can see it killed. So there is no more antibiotic. So you will get the resistance. Okay, by producing enzyme that inactivates antibiotic. As I said, bacteria producing beta lactamase enzyme, right? So in the secondary metabolites, as a secondary metabolites, which are those strains? Staphylococcus aureus, gonorrhea, and gonorrhea influenza, that produces beta lactamase enzyme. So what happened? This lactamase enzyme will act on beta lactam ring of the penicillin, right? Different antibiotics have the beta lactam ring. So that beta lactam ring will be cut, definitely inactivated using the beta lactamase enzyme. So your antibiotic will no what working, right? Now inactivation of chloramphenicol. What happened? It will be inactivated by the chloramphenicol acetyl transferase. It's called the CAT gene, CAT, C-A-T, CAT, right? Chloramphenicol acetyl transferase gene. So this chloramphenicol, this CAT gene is produced by the bacteria, right? And it inactivates the chloramphenicol. So your chloramphenicol will not working anymore, especially gram-negative bacteria. Uh, uh, Enzyme present constitutively, hence higher resistance. So generally you will get the higher resistance in gram-negative bacteria whenever you use the chloramphenicol. And in gram-positive bacteria, yes, we can induce easily. Inactivation of aminoglycosides. Inactivated by acetyl phosphoadenyl transferase present in gram-positive and gram-negative. So different group. See, you have to remember in gram-positive and gram-negative bacteria, different group of acetyl phosphoadenyl transferase enzymes that will inactivate the aminoglycosides. Now, alternative pathways for metabolomic growth requirements. Obviously, the bacteria who have the resistance, they are not following the routine pathways, right? They follow the alternative pathways. So resistance can also occur by alternative pathway by bypasses the reaction inhibited the antibiotic. So whatever the antibiotic reaction is there, so that will not follow that pathway, right? Where the reaction will happen, they will use the another pathway. So bacteria are very smart, you know. Okay, so mechanism of resistance. See, we have seen the mode of action, right? How it acts, that it act on cell wall, it act on 50S, it act on 30S, it act on cytoplasmic porine channels. So that was the mode of action. Now mechanism of action, right? So if we talk about the penicillin, it produces the beta lactamase enzyme, right? It cleaves the beta lactam ring. Now aminoglycosides already know. It modified the phosphorylating, adenylating, and acylating enzymes, chloramphenicol, CAT enzyme, right? CAT enzyme, chloramphenicol acetyl transferase, it inactivate chloramphenicol, erythromycin, change in receptor by methylation of rRNA. Methylation is a process, you know, I think DNA methylation and RNA methylation, it's called the epigenetics. So this is the different types of life sciences field. We were in 12th standard, you know only biology, right? When you grow SY, now I think you are TV, you come to know there is a microbiology, there is a biotechnology, there is environmental science, zoology, botany, there is a genetics. Yeah, but in the biotechnology, you will find the genetics. But in the field of genetics, there are different types of genetics, right? So one part it's called the epigenetics. Epigenetics means there are certain there are the role of certain molecules to play with the genetics. Actually, genetics is there, your gene is there, right? But gene is your, you can say, the you can say your fan. I'm just giving the example, your fan, right? The fan in the room is the gene, but the switch of that fan, it's called the epigenetics. So your gene, obviously, you will play with your genes. Anything with your gene happened or any mutation happened to your gene, it will deteriorate your life, right? But the switch is there. Someone is playing with your gene, right? So that switch is called the epigenetics. So certain factors are there that play with your gene, it's called the epigenetics. So one of the epigenetic part is a methylation. Methylation means whenever you add the CH3 group to your gene, to your RNA, it's methylated, right? And it will change your DNA, it will change your RNA. So here we can see the methylation of rRNA. So change in receptor by methylation of rRNA. So whatever the receptor is there for the drug, it will be changed by methylating the rRNA, right? Tetracycline, it will obviously reduce the uptake or import export, the balance of the import export, the disturb balance will be disturbed. Sulfonamides, active export out of the cell and reduced affinity of enzymes. Sulfonamides using the all the porine channels and cytoplasmic membrane, so that ABC transporter system will be destroyed or it will get imbalanced. Okay, now it comes to the quorum sensing. As I said, quorum sensing. Quorum sensing means two bacteria are talking in between, but it's not enough. On one bacterium, they are sending some signals or they are secreting some molecules that are very low in concentration. So it can't get heard or it can't get smelled by another bacteria. So numbers of bacteria will come together. They will secrete some signals, and in the large quantity, if that signals can quantify, then only another group of bacteria or same bacteria can hear that signals, or that signals, that molecules that can lead to some pathways, right? And it will help for the transcription or translation or replication. It's called the quorum sensing, right? In the layman language, bacteria can talk and can sense the molecules. So microbes communicate with each other and exchange signaling chemicals. It's called the autoinducers. So bacteria generally, they produce the signals. It's called this inducers. It's called the autoinducers. If I have to induce myself, right? Say for example, I have to write a paper, or I have to go for a drive. So I have to induce myself, right? Then only I can go for the drive. The same thing, if bacteria want to do something, they have to induce themselves, right? If bacteria want to replicate, bacteria want to transcribe certain genes, they have to induce themselves. It's called the autoinducers. These autoinducers allow bacterial population to coordinate gene expression for virulency, for conjugation, for apoptosis, mobility, and resistance. So what are the different functions of the quorum sensing? When any molecule secrete by the bacteria in the group, right? It's called the autoinducers, and these autoinducers can sense by the bacterial population for the gene expression of virulence, for the pathogenicity, for genetic transfer, for the programmed cell death, and for their mobility, and especially for the antibiotic resistance. So why it named as a quorum sensing? Single autoinducer from single microbe is not capable of inducing any such change. As I told, that single bacterium is not enough, right? So all the group of the bacteria, they have to come together and they have to secrete the molecules. It's called the autoinducers, right? So it's called AI1, AI2, AI3, right? But when its colony reaches to a critical density, now group of the bacteria, say for example 2 raised to 2 bacteria, or 2 raised to 5 bacteria, or 2 raised to 10 bacteria, it's called the group of the bacteria, right? So that quantity is called the quorum, right? QUO, quantity means quorum. Threshold of autoinduction is reached and gene expression starts. When any threshold of autoinduction is there, means so many autoinducers can be produced by the group of the bacteria, then only the gene expression will be started. So quorum sensing signal molecules are different types of auto-induced phosphatase, autoinducers 2, autoinducers 3, numbers of autoinducers are there, have been identified in gram-negative bacteria. And autoinducers 2 quorum sensing system is shared by gram-positive bacteria as well. So quorum sensing inhibition, what's that? So it is proved to be a very potent method for bacterial virulence inhibition. So if we want that bacteria should not be pathogenic, right? So it is very important method, that's quorum sensing. So several quorum sensing inhibitors molecules have been synthesized. Now molecules that are quorum sensing inhibitors that can be synthesized in lab, and these inhibitors have been synthesized and have been isolated from several natural extract, right? Say for example garlic extract, that's why garlic has the potential as antibacterial activity, right? So it is a, that's why they synthesize the molecules, that's called the QS inhibitors. And this inhibitor have shown to be potent virulence inhibitor both in vitro and vivo using infection animal models. So when you synthesize such molecules that can inhibit the quorum sensing, right, means what happened, whatever the bacteria producing the signals, that it will inhibit, right? So it will inhibit means bacteria will not grow anymore, it will not transcribe, right? So quorum sensing inhibition is very much required for uh, to stop the infection, right? So that is the alternative. See, if the next era we can say, if we don't have antibiotic, and pre-antibiotic era, if we talk about, if we can inhibit the quorum sensing of the bacteria, definitely we can achieve the we can achieve the antibiotic resistance mystery, right, what we are facing nowadays, because the results are very promising for in vitro and in vivo. Okay, what is the strategy to contain resistance? I think to solve the resistance, we can develop new antibiotics and judicial use of the existing antibiotics. We should not use the randomly antibiotics, right? Yes, 15 antibiotics from whole the spectrum. We can see only the 15 biotics from 167 under development had a new mechanism of action with the potential to combat the multi-drug resistance. Nowadays, only 15 antibiotics is working for the antimicrobial resistance, or we can say multi-drug resistance. And lack of incentive for companies to develop antibiotics. We don't have financial budgetary system, so we can develop new antibiotics. Okay, alternate approach, what we can, the alternate approach, as I said, quorum sensing is the alternate approach, right? One more alternate approach is the phage therapy. You all know what is phage. Bacteriophage means the phage can kill the bacteria when you use the Ganges water, right? Ganga, it has the bacteriophage. So that's why when anybody died, right, they can we can put the 1 tablespoon of water in his mouth. Why? Because it is having the bacteriophage, and yes, the bacterial it can inhibit the we can say bacterial replication, or it's like growth, bacterial growth, right, to the dead body. That's why we are using the Ganges water. So bacteriophage, it that can kill the bacteria. So phage therapy, yes, if we can use the phage, we can definitely overcome for this multi-drug resistance or antibiotic resistance. So phage therapy is the therapeutic use of lytic bacteriophages to treat pathogenic bacteria infections. Bacteriophages are viruses that invade bacterial cells and disrupt bacterial metabolism and cause the bacterium to lyse. There are two types of cycle you must be knowing, right? Lytic cycle and lysogenic cycle. Lytic cycle, in lysogenic cycle, bacteriophage can survive into the bacteria for years and years. And in the lytic cycle, bacteriophage will enter into the bacteria and it can kill the bacteria, right, and releases the phages, numbers of phages it can releases, right? So we need to go for this lytic cycle, and we can induce the lytic cycle artificially, and we can use this phage therapy. The success rate was around 95% with few gastrointestinal or allergic side effects. British studies also demonstrated significant efficacy of phages against E. coli, especially Pseudomonas species and Staphylococcus aureus. And Staphylococcus is the most pathogenic strain, and they are getting the 95% of the success rate using the phage therapy. So it's a good alternative approach to treat the antimicrobial resistance or multi-drug resistance. Some newer antibiotics already it has been developed, and the mode of action and mechanism of action is totally different. And yes, let's see, linezolid that can target 50S, and tigecycline that can target 30S, streptomycin. It can depolarize the membrane and can enter, and the elbevacine inhibits the cell wall synthesis. Telavacine, inhibition of cell wall synthesis, and it can disrupt the membrane, and ceftriaxone, ceftaroline, and the cephalosporin that can inhibit different types of enzymes. We should avoid the random use of antibiotics. That's it, right? We cannot judicious use of antibiotics should be prevented. So contamination of antibiotic resistance is multi-pronged program. Yes, everywhere it is going on that we should not contaminate our body with random use of antibiotic, right? Especially involves all stakeholders, physician, patients, and pharmaceuticals. It's a business, totally business. So we don't have to, we have to be very careful while using the antibiotics. Now, as we talk about the different types of factors that can affect the antibiotic resistance, the drug-related factors, we all know environmental factors, prescriber-related factors, and patient-related factors. Yes, anyone can't prescribe the things, and we cannot take the antibiotics without prescription. Drug-related problem is there, environment-related problem is there, right? So tomorrow we will go one by one. What is the environmental factors and drug related factor? I think it's enough for today. I think not tomorrow. Where is my next lecture? I think on Wednesday. Okay. Let me see. How many slides are there if I can continue? I think in the next lecture, one by one, we will, I will explain the different factors of the antibiotic resistance, factors affected, and what should be done and not should be done, right? So till then, you can go through the slides, you can go through the literature. See, I have shared one research article on your portal. You must have to read that antibiotic past, present, and future. And we will discuss this research article, right? So everyone must have to go see. Only PPT is not the things you can just read it and you just prepare and you can come to the exam, right? You have to go through the books, you have to go through the research paper. You can go for available all types of materials. You can see the YouTube videos, right? You should have more knowledge than me, right? So next time when you come, please go through that research paper, a review paper, and we will discuss. Okay. Okay, till then, bye. Have a nice time.